a bit of a historical overview. It's like a bit of like protein structure prediction from the last, well, mainly the last maybe 5 years, but like also a bit of history of earlier things and what I think they are the key concepts that people have. I mean that was important for the development of AlphaFold. So as I said, just if you have some questions or anything that's unclear, just interrupt me and I will try to tell you. I will try to explain it better. So make this once more, otherwise I can see. Protein structure prediction or understanding protein folding has been an important concept to understand, try to understand it since at least the 1970s. So of course the idea is sort of like we know that proteins, most proteins, not all, but in general proteins have a well-defined three-dimensional structure given a certain sequence. So basically if you have a sequence, whenever that is expressed in a cell, it folds to something that is roughly one structure. There are variations of that, but in general that's true. And of course that means that there is some kind of landscape or some kind of funnel shape that sort of guides this folding. So people have studied this for years, and people have realized that it's, of course, you cannot search all conformations because the conformational space is too big. And at the same time, the difference in energy between the folded state and all the unfolded state is not gigantic. It's maybe some tens of kcals or something like that. So it's so if you heat up a protein or if you change the pH, something like that, you can get it to unfold. But then in many cases, you can fold it back. So this was Arne did in experiments in the 1970s. And of course, already since we had a few structures, people tried to basically say, oh, if this happens in nature, we should be able to do it in the computer, we should be able to do it. So one option that people done is to try to describe the energy of a system. So people try to do it either with more physics-based or more knowledge-based, but more statistical potentials. So basically tie how likely, what is the cost of having a hydrogen bond formed, another hydrogen bond form, or water in a certain position and so on. And you can do that, and you can do, with the physics-based one, you can set up something that you can make that quite accurately describes the energy of a system based on pairwise interactions. Basically, every pair of atoms you have to take into account. But you also have things like the backbone conformations and all the side chains that are important. You probably do not need to take into account triplets. There might be some need for that for polarization effects in water, but you can probably get around it. And you don't need to take into account quantum mechanics calculations. At least that's the general idea. The problem is like if you do that and you can run a molecular dynamic simulation, so you basically just take the derivatives and you iterate over a certain time step. So you start from one position and use just Newton's second law, and then change the motions. You can simulate the motion of protons, but it's just that the folding time is many orders of magnitude too long for us to be able to do that for most protons. You can do it for some very fast folding small protons. You can do it with special big computers, and it sort of more or less works. You can use a bit faster knowledge-based method with coarse-grained descriptions, but then you lose accuracy. So people have tried to do this since the 1980s or whatever, and so far it has been quite unsuccessful at predicting structures of proteins, I would say. So what people did, one problem is like the other people was trying to divide proteins into smaller fragments and look at each fragment at a time. So the problem here is like if you take these two fragments, so as you see, this bluish beta sheet here and the red alpha helix, the sequence of these two fragments are identical. So they're the same exact sequence, but in one case, but the structure is very different. And so if you want to make a predictor of even the secondary structure of these cases, that predictor is gonna, if you just have this information, you're gonna do the same predictions in both cases. So if you do that, it doesn't work. It doesn't know what to predict, so it can't work. However, if you add evolution, so if you know that it's also known that two proteins that are evolutionarily related, so proteins are called homologs, they have a common ancestor, either in the same species or in different species, or in ancestral species. They often, almost always, share a similar structure. So basically, if you have many proteins that are evolutionarily related, we can assume that they all have the same structure, and maybe that provides extra information. So this is what people did in the 1990s, and for instance, for the secondary structure problem like that, if you have a problem with this sequence, if you have the sequence and do the secondary structure prediction. Was it the question?
I thought I was saying the community got the last one.
Sorry about that. I'll mute the.
Okay, sorry, that was not related to So if you take this sequence, you do a secondary structure prediction in these cases, you see that in the middle of this is going to be sheet in both cases, and then so the prediction is going to be correct in one case and basically wrong in the other one. However, if you add this evolutionary information, which you do by searching a database of sequences, and then you get a multiple sequence alignment that basically tells you what is the average or what is the conformational variability in each position in a protein. You can do that, and you create what you call a precision-specific score matrix, basically probability for each position, each position in a protein. So like in the green position here, it's always a green amino acid. In the blue-yellow one, it's either blue or yellow, or in this one here, more Jamaica colored, it can be anything basically. So you have some positions are more conserved, some positions are less conserved, some have some specific types of amino acids and so on. And if you use that information, you can improve the secondary structure predictions by quite a lot. So you can do that by doing a proof. I think this should be next slide. No, it's not. Okay, well, yeah, well, okay, that slide is missing here. But then you can prove the secondary structure prediction. So then you can predict, and this is what Burkhardt-Ross people did in the 1990s, and that improved secondary structure prediction to something like 70% accuracy or something like that. So and since then, maybe better methods has pushed a bit further, maybe to 75, 80%. However, this is still just the secondary structure. So if you want to predict a structure, you need to combine this sort of prediction of secondary structures or fragments with some algorithms that put them together. So this is what David Baker primarily did starting in 1997 and then kept on doing that for the next 20 years, or at least the next 10 years, using the Rosetta program, and other people did similar ideas, like David Jones had FragFold, and Jim Bowie had methods for this also. So the idea is then to basically use this secondary structure prediction and other methods to then identify fragments. So you have a list of possible fragments for each part of the protein, and then you use some kind of knowledge-based folding potential to put them together. So like this is sort of a simulation here. And this sort of worked in some cases. It was never very reliable, but if you did it carefully, you could actually predict the structure of proteins. But it was not generally applicable. It was slow. It worked better for alpha helical proteins, for beta sheet proteins, and it only worked for small proteins. And the accuracy was not very high. So that was what, I guess, the most successful method until 2010 or something like that. And then people realized, or actually already before, people realized that this multiple sequence alignment contains more information. So the idea is that before we only looked at the average information for each position, each column basically. But you can also look at the information. So in these two positions here, you see that you either have blue yellow or yellow blue. So there you have a co-evolution. You may either have a big amino acid in a small, or a small and a big, or a positive and negative charge. It means that these are correlating with each other. So you have a correlation between the pairs. And of course, you can calculate the correlation between every single pair of amino acids. And the first papers that tried to use this information for protein structure prediction appeared in the mid-90s also. One was by Alfonso Valencia and Chris Sanders, but there was also a couple other papers. However, the method didn't work very well. It was like, it was better than random, but it was not very accurate. And I will come back soon to why it didn't work. But here's a link to a lot of papers by Sergei Shinikov that keeps tracks of all these papers. So the idea is, of course, that if you have this co-evolution, that should relate to amino acids to be in contact with each other. Because like if they're closer together, they should correlate. So if you change Yeah, the idea is here basically, if you take a protein, if you look at the detail of protein in the interior, it's often well packed. So often it has a nice packing. So there's not room for a lot of water, a lot of holes. It's a well packed, nicely folded thing. However, sometimes in evolution, you have a mutation. So in this case, the pentamer here mutates to an arrow. And of course, this mutation will make that the packing is not as ideal, but maybe it's not too bad, so maybe the organism still survives. However, there will be a preference for having a compensating mutation. So you have a second mutation that compensate for this loss of interaction. So you have, in this case, a quadrate of mutated pentamer. And you see the packing is nice again. So that sort of explains, like if you have one correlation, you have another one. However, there's a problem here. Like if you look at the top of this arrow here, that part is not identical as it was before. It has changed. So this could even be correlation with that position. So that means you have a network of correlations that are coupled to each other. And of course, because everything is coupled to everything else, and if you look at the big multiple alignments, you will have big networks of things that couple together. So you have both direct and indirect couplings. So these indirect couplings is something that made this prediction of contacts with these coevolution signals. If you don't take that into account, that you do much worse predictions. And that was a few papers, 1999, 2000, particularly by Lapidis, but they were sort of they were sort of ignored because, the reason was because they were not very practical, because you needed very big multiplex alignments. And in 99, that wasn't available. But the idea was there, and then it was sort of reinvented by Martin White and others in 2008 and so on. So the idea is basically you take an MSA, you look at two positions here, you calculate the column. Well, I can skip this. Well, okay, go ahead. So this direct coupling methods, we call them, was developed. The ones that are used later was developed around 2008, and there was a few different methods that were improved a bit up and down. But it's the popular one called DCA, direct coupling analysis. So the idea, if you look at this as an example, so this is a classical way of describing contact maps. So you take your protein here to the right, you have 250 residues here and 250 residues long, and you have basically, you plot the sequences at both X and Y axes, and you have a gray dot here whenever these two residues are in contact. So in contact in these cases means that they are less than 12 angstrom away from each other or something like that. And then you have, in this case, you plot the predictions. So here we have a method called mutual information that just takes correlations. And the blue and red dots here are the ones, contacts and predicted ones. Blue are correct and red are wrong. So you can see that most of the predictions we do there are wrong predictions. And you can see that most of them are close to, like, clustered to close to each other on the diagonal. So diagonal for things that are close to each other in a sequence. So you can understand that if you take all these red and blue dots and try to fold the protein, it will not be very successful because you don't have the information about it. Although there are some contacts, it's better than random. If you take random, you will only have 1% correct ones. Now we have 22% correct ones. So it's not completely useless, but it's not good enough. If you use this method, or use one method called, two different methods, one is called PLMDCA, one is called Psychov. And in both these cases, they have about 50% correct, 52, 58% in this case. So these are the methods that take into account these indirect couplings. And you can see that you have contacts like here that are very far away from each other sequence, between like residue 40 and 250, that are correctly predicted. Of course, there still are a number of contacts that are wrong, but you can imagine that if you have all these contacts, you can actually possibly predict the structure of proteins. And that was shown that you can actually do that to predict the structure of small proteins. And and then we showed that you can actually improve that by just using some simple machine learning. So the reason is, of course, if you think about this as an image, is that these dots are certainly not randomly distributed. You see that there are a lot of patterns here that you could learn. So, and of course, learning patterns is what machine learning is experts at. They are very good at math patterns. So in this case, we had a very, very simple machine learning method that basically tried to run a different set of different things and so on. And just look at the small arrow, and you can see that we could improve this prediction quality from 58% to 70% in this case. And you can see you get rid of quite a lot of like small random dots that are not appearing here where there was a random one. And you sort of get stronger signals for areas that you missed otherwise. So a combination of these TCA methods of machine learning could improve the predictions. And that's what many people did for last year. Skip this. So we did something called our second version, Pcons C2. So that was our first attempt to sort of do deep learning. So we took these contact prediction methods, we did 2 different methods, and we did different alignments. We had like 8 predictions. And then we had a deep learning framework, which was in this case just based on a random forest, but iterated through it many times. So we call it that, and we added some other information. So basic, I think that's next slide. So in the first layer, we only had like 36% correct predictions. But then we used this as an input to our machine learning method and predict the next layer, and we get up to 50%. And then we at the end of the 5 layers, we got up to 70% correct predictions. So basically, we could get rid of single predictions that were like sporadically appearing, because most of them predictions should be close to other predictions. So we sort of strengthened the prediction that was close to other predictions. We still miss a lot in the beginning, and we have a few false positives, but at least the predictions got much better after 5 iterations. So this was our attempt in 2014. Yeah. And then people, we and many others improved this method. In particular, it was a paper by Jimbo Xu that used what's called ultra deep learning model. That was a clear improvement over early methods. And it's basically quite similar to this is what they did. So that was really if you remember, or if you know that the history of deep learning was in 2012 was the big success in ImageNet. So they used the same ideas of people use for images, but to predict context. So you basically took your predictions here, which was this L by L, so L predictions, and then you add to that, you add something that you predict on the sequence. So you take your sequence profile and you predict secondary structures, whatever, and you have a rest-of network to predict something. And then you can go from a one-dimensional L network to an L by L network by just doing an outer product. So yes, you know the product here, and then you have a number of layers here that predict. You merge them together, and then the whole. So you have an L by L by some number of colors network, and then you do a 2D convolutional network as deep as you can, and you predict contact map. And so basically you start with some sort of contact map. We add information from the sequence that you add to it as an extra layer, and you can build the contact map. And that was a clear improvement. So this was an example of a prediction in CASP 12, I guess, or CASP 11 maybe, where they use this method. So you can see that the red one is the native structure, the blue one is predicted one, and they are quite accurate. So this was a success in this case. We did something similar. We did our false version. This one was developed more or less at the same time. We used something called UNet here that we did the same thing. So we had a prediction of secondary structure, sequencing of that. We did the outer product. We merged them together with predictions. We had this UNet that sort of processed. So it was not the RESTnet, it was a UNet that processed the network, and then we predicted this. In this case, we predicted both the contact maps and actually also the distances between each residue.
Professor,
so, yeah.
Just a question here, and I think about the previous slides. For both of these 2 models, the UNet and this one, I think the slide before, where yes. I've been thinking about the result that we want to predict is the contact map, how close these two, the antigen and the antibody, can come close.
In this case, it was not antigen antibodies, but it was just individual proteins, yes.
Okay. Yes, the question is about the inputs into this model. What can be said, or how can we reason about the input data in both these two models?
So the input in this case is, the input in the model is actually these predicted contacts. So the input is these DCA predictions. So basically this type of prediction maps. So the input plus some extra information, but that's, so basically they use this PLM DCA or something like that as an input. So they do not handle the multiple sequence alignments directly.
Okay.
So the input is
So
the coevolution and information per potential. This is the input here. So in our case, the input was this DCA predictions here in a few different flavors, but basically the input is basically this PLM DCA plus some extra information, but that's marginal information.
Okay, makes sense. So the
Okay.
result still, the result out of the model would be still
A contact map, basically, saying that the residue
contact
I and
map.
J are in contact or a distance between them or something like that. Yeah.
Okay. So someone is asking, the co-evolution analysis seems crucial for the contact prediction model. How does the model distinguish between true co-evolutionary signals, which indicate direct contacts, and indirect correlations that might arise from a network of interactions?
So the way that this DCA method solve that is basically by having an underlying model that it tries to, so you have an observation, which is the correlations, and then you try to basically have an underlying model that sort of explain that. So you basically, I mean, practice what you do is you actually invert the matrix. That's what you need to do. But it's, the ideas are from, you need a model that sort of like, you know, if A correlates with B and B with C, what correlation do you then see with A and C? But there are a couple of, as a practice, you end up inverting a matrix. That's what we need to do. But the ideas come from spin glasses originally. So this is like 20 years old things that are done by physics and spin glasses. There are some other methods also, but the one that dominated was this DCA methods, that is basically in a matrix inversion. But
Yes,
we will come to how AlphaFold solves that later because they ignore it. That's I think, yeah.
Prof, one more question. I think
Yes.
for since we are a multidisciplinary group.
Yeah.
For computer science, one would wonder what can be reasoned about a contact map, or what is a contact map in this context, but also what can be said about the contact map in terms of the antigen antibody or in terms of the sequence structure of these two.
So in some examples, the contact map is basically just like here. This is the definition of that this residue here and this residue there are within some cutoff. So they are within 12 angstrom or whatever cutoff you want to do. So this is just saying that these two are in a certain cutoff from each other. That's the definition. So, and of course, from in a structural perspective, you can imagine if you know all the contacts. So this is a two-dimensional. You know the structure of the protein because you basically have it well defined. It's like I mean, you could think about, in theory, you could make a mirror image of the protein. That would be identical because of course the contact doesn't know if it's which direction, but there are other things you know about proteins that's impossible. So that's, but otherwise, it's information is really the structure because that's, I mean, you could think about something that is like very long and bent and have an identical contact map to something that's very long and straight. So then you maybe not have the information, but that's very rare cases. But in general, something globular that really tells you the whole structure. So it's just very much another representation of the structure.
Perfect.
Which, except for mirror imaging, it contains all the information.
Thank you.
Okay.
Yeah.
So what we show them is that one problem with these same methods was that for them you needed very big multiple sequence alignments. You needed thousands of sequences and so on. So and that's the reason why these methods didn't do very well in the early 2000s because that was rare. So here was a plot that we did against the size of the multiple sequence alignments, or we call it's a bit filtered, so it's called effective sequences. So it basically takes away things that are too similar. And if you look at these DCA methods, there's all these dashed lines here. These are different DCA methods. And you see, if you have only 100 sequences, they have an average accuracy of like 15%. But if you have a few thousand, you have an average accuracy of 50%. And you need probably something like 50% to do a good model in this case. So like you really see that it was increased a lot with size. And the red line here is an old method that was developed that does not use this coevolution signal, but use machine learning only. And then you see that is quite flat. It really depends not very much on the size of the multiple sequence alignments. So for small multiple sequence alignments, it's much better than the DCA methods, but it never gets more than 30% accuracy, basically. There's a small dip here around the 800 or 500. For some reason, there are some difficult cases there. I don't really know why, but there are some pairs that are difficult. So that's difficult to know. And then you see, this is different versions that sort of combine machine learning and this sort of DCA methods. And you see that these methods are always better. And even maybe a few hundred sequences, you get to an accuracy of 50%, 60%. So you have good enough accuracy to predict the structure. So it really shows a combination of these DCA methods and machine learning was very beneficial for these predictions. And this was just an example in CASP level 12 that we did a good prediction. The gray is our predicted structure and the black is native structure, and they are almost perfect for one case. And it wasn't best in every cases, but there were some few cases where it was good. And we developed the automatic pipeline that could do this. We took it, starting from a sequence, we could run these different methods, different prediction contexts, making contact maps, making folding the structure, and making the models. So we could do that, and at the end, and Sergei Shinnikov and David Baker did the same thing. And at the end, we run this for all protein families that had no structure. It was something like 7,500 families at that time. And for this, you could make good models with something like 500 models. And they did another 500 models, and the overlap was not that big. So maybe for 10% of these families, we could generate good models that nobody could generate before. And in the cases where the models were similar, at least the models were similar. So that was not identical, but it was similar. So it was quite good to do that. So we could basically add 10% of the missing families we could have models for. So we could do that. But of course, it was not for everything. And then we entered the days of CASP 13. So CASP, I think, we can actually show this one. So CASP is this competition. It's not called competition, it's called assessment. So a critical assessment of protein structure prediction. So that's a conference that has been running since 1994, mainly organized by John Molt, who where everybody can compete in predicting the structure of proteins. So there are about 100 targets every year, and there are 100 targets that no one knows the answer to. So there are new protein structures that are going to be solved. So there's no way of like cheating or like overtraining methods. Actually, these are novel, and they're novel for everyone. And you can divide these targets into easy to difficult, mainly based on how similar they are to proteins that are already known. And this has been done since 1994. So in 1994, you have a dark red brown line here that sort of shows the difficulty. So even then, and this is the performance, I think, the best groups every year. So even then, for easy targets, there were some good predictions. So this is measured as EDTTS, which is like 100% is perfect, probably 90% is close to exponential accuracy. So you see in 1994, for easy targets were good models, but then more difficult targets you got, the performance dropped quite significantly. And then you can see already in CASP 2, you did an improvement of the hard targets, and then you see that from CASP 2 up to CASP 12, there was, in particular in the medium hard targets, there was a linear, constant improvement. For the hardest one, was very small improvement. But it got a bit better and better for the more difficult targets, but not for the most difficult ones, or only marginally better. And then suddenly in CASP 13, DeepMind introduced AlphaFold 1, and there was a big jump in performance. So suddenly we got up to 60% EDTS where early methods had more like 30% or 40%. So it was a big jump in performance. And you could see that they also predicted contacts. That was in this case, like one part is this is the correct contact prediction, and this is the native one. But it's not contacts, it's actually distances. And you can see that they are almost identical. So this is a number of targets with both sides here, not perfectly identical, but very, very similar. So they could actually predict the distances between every pair of residues very accurately. And this is just some examples of the models. So you can see the models are almost perfect. And there are, see here, part of the middle edition, they're not really perfect, but some of them are really, really spot on. So much more higher quality than anything else I've been on before for these difficult targets. And the idea, now this is not this is better slide just. So the idea is not that different from people done before. So basically the idea is basically you take the sim, you take these multiple six alignments, you calculate this PLM DCA, these coevolution couplings and scores, and then you do a 2D convolution network. And then you predict not distances, but you predict distance probabilities. So you have a max sum probability of 0 to 20 angstrom, and you have a max sum probability of that one. The difference is, I would say, is mainly because they had a much deeper network than people had done before. And one way they managed to do that was to not predicting the whole contact map at once, but predicting it or training it by 64 by 64 or 128 by 128. So predicting on like chunks of the network. And then they did that and then they took these predicted distance probabilities and made some kind of energy potential from it. So basically had smoothed this function and made basically probability distribution for every pair of residues. And they put that into a very simple machine minimization program that just made models, and then they add some sidechains on top of it later. So that was quite successful. It was clearly a step forward. So people were a bit shocked, but it was not nothing fundamental new, but it showed that we are doing progress. And of course, if you go back to how it was a few years before, progress had been quite limited in the last few CASPs. So people were quite happy to suddenly see big progress. So we and others sat down after CASP 13 to think about what is next. Actually, for us, it was clear that this deep learning methods is better than anything else. They're better if you do it correct. It was clear that most single domain proteins can be predicted quite accurately. So if you have a simple protein that has one domain, it is actually, in most cases, you can get a good model of it. And it's also clear that the next big challenge is not going to be single proteins, it's going to be protein-protein interactions. They can be clear and important. So we set out to work for that. So we basically, okay, skip this. We did the same thing. So, we took the same ideas, but instead of predicting the structure of a single protein, we want to predict the structure of protein pairs. And in principle, it's identical. There's only one problem you need to solve. You need to match what line in each MSA corresponds to the interacting pair. So, of course, you want to calculate this correlation, but you don't know exactly which protein is on which line, because they're not going to be the same thing. And as long as you have, if you have a number of species, number of bacteria, and you have one copy of protein A and one copy of protein B, and in all these organisms, then it's quite easy. You can assume, okay, in organism A, these two proteins interact, in organism B, these two proteins interact. However, this is not always the case, because in many cases you have many homologs in one species, but not in another species. So you have to sort of try to see if they all interact, if there's a specific pattern to interact and so on. And normally the only thing you can do is basically take only like the most similar pair in each organism and assume they interact in the same way. But that means that you throw away a lot of information, which makes these methods less accurate. So we developed this method, and we call it fold and dock. And so we took a couple of MSAs, MSA1 and MSA2, we put them together. We used this program T-Rosetta to make the distance predictions. We used PyRosetta that we hacked a bit to both fold and dock this program together again. So earlier methods that have been trying to put predictive interactions took just a single structure and put it together. However, here we allowed flexibilities. We allowed them to change structure and all. So we both folded both processes and docked them together at the same time. And then we had a scoring method that basically was a consensus-based scoring method that sort of like that if you run this many times, you get the same answer every time, you're most likely correct. If you get different answers, you're most likely wrong. So we did that, and in some cases, it works very well. This is a prediction of one protein. And there is actually 2 green and 2 blue models. You can see them if you look carefully, but they're identical. And you can see the contact maps on the upper and lower part are almost identical. However, in many cases, it was not as good. So this is the case with the failed OR. Each individual protein is nicely predicted correctly, but the interactions are not predicted correctly. So this is a case of, that didn't work well. So at the end, it didn't do much better than other methods. We made a few good predictions and we could do something about it, but it was not a huge step forward, but it was another approach that sort of worked, that looked promising. And then we ended up in 2020, and there was pandemia, and there was CASP-14, and we got to see this plot. So if you remember, we had a big jump from CASP-12 to CASP-13, from the red to the other red line here, the thick red line. And then this time we had another big jump up to the black line that was basically flat. So basically, even for the most difficult targets, we had prediction that was close to experimental accuracy by one group, and it was DeepMind and AlphaFold 2. If you take all the other groups, the best people did something like the dashed black line here. So that was basically the best methods that without DeepMind. And they were basically as good as DeepMind was 2 years earlier, or slightly better maybe, but not much. So we were, of course, quite shocked. I mean, we were sitting in our own universities, homes in pandemia, and we tried to understand what people had done. So we could see that it was, but this is a smaller plot showing improvement, basically that it was a big jump. And you can look at here, you can see some of the models. You can see this is like predictions are, this is an example in figure B here. AlphaFold is blue, experiment is the green one, and they are really, really identical. The RMSD is 0.8 angstrom. So every single atom is basically perfectly plotted. Look at this plot here on the medium error RMSD. It's a medium error on positioning of 95% of the C alpha positions. It's like less than 1 angstrom. Or the second best method had almost 3 angstrom. So it's an order of magnitude. And they're also going to do predictions of very big complexes, like here to the right. It could predict things like a zinc finger binding region. Although there was no zinc there, they know about zinc, but they had a perfect prediction of the zinc finger binding. So even on the atom level, on the residue level, it was almost perfect. And and this is how AlphaFold 2 worked. And I will go through this in a bit more detail because I think it's interesting. So we can start here to the left. So you start here with a sequence. And as before, you do the database search, and you get a multiple sequence alignment. So this is not part of the machine learning part. This is standard. You can also add templates and pairwise information, but let's ignore that for a moment because it's actually not that important. And you can do this get this MSA. But before, people used this PLM-DCA or Psychov or something to calculate the contacts. Now this is completely ignored. They just take MSA as it is and put that into the machine learning pipeline. And from here on, everything until you generate a structure is coupled. So you have an end-to-end learning. You're really doing machine learning from the beginning to the end. So that means that if you have some errors here on some position of an atom, that's going to affect how you calculate, how you extract information from the MSA in step 1. So because you have a derivative, you can do backpropagation. So normal machine learning. And that's part of the explanation, but probably not the most important part. But anyway, you can extract information from this MSA. You use something called the EVA former. That is 48 blocks. I will tell you more about that in a second. And then from that output of these 48 blocks is a pair representation. This is sort of a representation of every pairwise distances and also the sequence representation. And then you put this into a structure module that actually builds the structure. It was shown in the ablation studies in the paper that this part is actually not that important. You can stop here and you get like 95% of the accuracy on that. So it's the key thing here is the EVA former. That's really what made the predictions very well. So let's look at the EVA former. So this is 48 blocks of things that are up, that are repeated. And you start with the MSA. And you have used, everybody knew about the paper that all you need is attentions. People use transformers and attentions. So here you have attentions both along the rows and then along the columns. So you have one attention layer in the rows and attention layer in the columns. And then you take this MSA, which is basically a one-dimensional representation. You do an outer product mean, and you get the pair representation. Of course, you merge it with your input pair representation. And here you use what they call a triangle update. So basically, you also have attention here, but you can do it not only on pairs, but on triplets. So you can have I, if I and J has a certain feature and I and K, that puts some constraints on J to K. So of course, if I and J is close and I and K are close, J and K can't be too far away. So you have triangle attention. And you have a self-attention update here also in the same way as you do an attention. And then you do another transition back to a pair representation back here, so you get an output that is another MSA. And you repeat this 48 times. I can skip the structure. And then you have a structure, and then after 48 times, you put in this pair representation and a sequence and a single representation there. You basically describe the protein with the backbone frames, and you do that as pairs of distances, or pairs of or not distances, but directions, and then you generate a structure, and at the end you predict side chains, and this is repeated 8 times if I remember correctly. So that this is mainly a quite fast way to build a structure. In AlphaFold 3, they replaced this with the diffusion model. That is probably faster, but yeah, that's probably the main advantage of it. It has some other disadvantages, but it's mainly faster. Skip this. Let's go. So we went on with this AlphaFold 2 when it was released and did the same thing as we've done before for predicting interactions. So we took 2 MSAs, we combined them. Instead of running TR Rosetta and DCA and those methods, we run AlphaFold 2. And to be surprised, they are, we got much better performance. So this is a measured performance. So if you took DocQ, which is a measure of how good the interaction is predicted, so like 0.23 is a good cutoff for being good and bad. So this is early methods, like including AlphaFold Doc, basically had, sometimes we had some good models, but on average, the models were really bad. In AlphaFold 2, if you just took it without doing anything else, we had an average score maybe of 0.1, but 44% of our models were higher than 0.23. So almost half the models were correct in some way. And then we sort of played around with these multiple sequence alignments and we get up to almost 60%. And the average score of 0.3 or something like that, which is unheard about in docking before. And we can mention that this version of AlphaFold 2 had never seen 2 proteins together. It was completely trained on a single proteins. So it had learned something more fundamental than about that could be used for predicting interaction also. Or in another way, the interaction of 2 proteins is quite similar to how a single protein looks like in whatever features you have. We showed that, we developed a scoring function called P.Q that could be used for predicting if the model was correct or not. It can also be used for predicting interactions, so if two proteins interacted or not. So it was basically based on the quality of the predictions together with the size of the interface. You can skip this. And yes, for feedback now, AlphaFold 3 is out, and the main difference is that you can handle other types of molecules than proteins also. You can have small molecules and RNA and DNA. And it sort of works a little bit worse on these things, but it's at least it can handle it. You can skip, it's based on similar. The difference is basically they spend more compute time on the structure than on the MSA, and they have a diffusion model at the end. So instead of AlphaFold2, you had all this EVA former that's replaced by the pair former in AF3, and the pair former does not use the MSA as an input; it uses only a single sequence. So it spends much less time on co-evolution signals and more on structure signals, and that's probably that's probably the reason why it works better for antibody antigens. But and then there are details, and we can skip the details here. So we bend well. If you look at for proteins, the difference between AlphaFold3 and AlphaFold2 is not that big. AlphaFold3 is slightly better in most cases, but not always. And if you run AlphaFold2 many times, something called massive folds, you can get more or less equal performance. So AlphaFold3 for proteins is just a marginal improvement. For RNA, it's also just marginally better than the best other methods, or quite similar, actually slightly worse than best other methods, but it's quite okay. This is here. I should have another slide. My antibody, sorry, was not here. What had them here? But it should be. This one I have it. There's antibody part. So antibody antigens, and this is work by my postdoc Samuel From, and we know that antibody antigens is a difficult case to predict because in this case you're missing a co-evolutionary signal. There's no co-evolution between an antibody and antigen because the antigen is fixed and the antibody is just evolving to fit there. So the antigen never changes. You don't have a co-evolution signal. So this is just an example of antigens. An antigen here, this is the purple thing in the middle, and then different prediction by different methods about that are predicted. And in this case, I guess the gray one is correct, and in this case, AlphaFold3 in red prediction here makes correct prediction. All other methods make bad predictions. And one thing that people have done before is that if you do many predictions, AlphaFold2 and AlphaFold3 are more and more likely to make a correct prediction, which is, of course, it makes sense. So if you take, in this case, we made 200 predictions with AlphaFold2 and 200 with AlphaFold3 and with a few other methods and a few different ways of doing that. And if you look at it, and then if you take the best prediction out of these 200 and you look at average docu, you got plots here to the left. And you can see the first thing is the red line here, AlphaFold3 is always better than AlphaFold2 based methods. And also Shy1 and Boltz1, which are clones of AlphaFold3. So it's something in AlphaFold3 that makes it better than the other methods. And you can also see that also if you look at the log scale, all methods basically is almost a linear, log linear increase in the quality of the best prediction on average. So basically, if you make 10 predictions instead of 1, you go up from 0.3 to 0.4 in average quality. If you make 100 instead of 10, you make go from 0.4 to 0.5. Of course, at some time it will level out because you can't go higher than 1, but it means that in a log scale, more predictions are better one. However, it doesn't mean that we do best predictions because if you make 200 predictions, you also need to know which of these 200 predictions is the best one. So if you instead use the top rank prediction, you can see that we almost flattened out of the 10 of 20 predictions, default is 25. So somewhere the increase from like 1 to 10 is quite okay. It's from 0.27 to 0.35, but from 10 to 100, it's only 0.35 to maybe 0.37. So we are not very good at finding the best predictions. Like we have a good models in many cases, but we cannot find it. So if we really lose a lot of performance by not being able to identify the good models. But still, AlphaFold3 is better than other methods, but all the methods behave more or less similar. Maybe Boltz is even more flat than the others. So that's one problem we have with antibody antigens. We can generate good models, but we don't know which one of it is. We can also look at, in the case we make good models, how similar are they to the training set? So the data set we use for training, which is all of PDB before 2021, or September 21 even. And if the similarity of the interface, I think this is less than t-squared less 0.6, you see that the red one is the AlphaFold 3 here. You see that there are a few models that are good, but most are bad. If the similarity between 0.6 and 0.8, you see that maybe half the models are good. And if more than 0.8, maybe 75% of the models are good. It's clear that AlphaFold 3 has learned something about data set that it sort of can memorize and then use for predicting new things. It's not something trivial because we are taking away all the trivial cases. And you see that AlphaFold 2 cannot do it. Shy1 has a similar feature, but not as good. So Shy1 has also learned something more about it. I mean, there's maybe some signal AlphaFold 2 also, but it clearly shows that it's probably not that AlphaFold 3 has not learned something new physics. It has learned to memorize the data set. And that's shown by that it does better things that are similar to the data set. And this is the typical example for each case. If you have maybe 200 models, there is a subset here of maybe 20 models or targets that always are good predicted. There is maybe 50 models that are almost always bad one predicted, except there's like 1 or 2 or a few good models here and there. And then there's another 30 models or 30 targets where there are half the models are good and half are bad. So like you can see the average is somewhere in between. And we still don't understand why this is the case more than the structural similarity. So these are more structurally similar to PDB, but exactly what it is that it makes this easy and this hard is a bit unclear to us. And this is just an example of when we do good predictions. So you can see that this is the same example as before. So like you see that the blue one is the native one, and the blue is AlphaFold prediction. The gray one is somewhere in between. But you can see that the most similar thing in the database has a sort of similar structure, but it's not identical. So clearly, what is it that is memorized in the database is very unclear to us. Yeah, this one we can skip. So that was basically, yeah. So in summary, we can see that AlphaFold 3 does a better job, but it does not by really understanding physics or anything better. It does it by some sort of memorization or training set, which maybe are not a bad thing, but it's at least is something that is it's different from understanding something new. It has some limits to it, and we can see similar trends for RNA prediction and also for small molecule predictions. So that is something that is similar. So yeah, so it's clearly it sort of spends more time on learning structure, but that results in more of a memorization structures. So that's that was my last slide. I have an acknowledgement slide here, I think. So this is Samuel did the antibody antigen work. Patrick was one of the ones that really developed the folding doc. Patrick and Gabrielle, they're not here. And then the other people are my current set of PhD students and contacts. So yeah, thanks for your interest. Do you have any questions?
Thank you. Of course, thank you, Prof.